I want to welcome everyone to another ALK Talk, and I'm going to launch a poll real quick. My name is Mark Rosenzweig. I'm from West Bloomfield, Michigan. It's my 12th year with ALK, and I'm a patient like you, and we're going to learn a little bit about some vaccinations. So we're going to go hit some polls here. What type of treatment have you been receiving currently? Share the results, and alectinib is at 72%, lorlatinib is 22%. And brigatinib is at 9%. And we have a few other people that are doing various other treatments. And I'm going to go to our next polling
Wait,
question.
Mark, hold on. I'm going to read the rest of them out real quick.
Go
We have
for it.
brigatinib at 9%, alectinib is 72, lorlatinib 22%, Nuvalent is 4%, or a clinical trial. Chemo with a TKI is 3%, chemo only 1%, gilteritinib. We're waiting, guys. We're hoping we're at 0 right now, but let's get that going. Thanks, Mark. On to the next.
Here we go. When were you diagnosed?
And I know these may seem redundant for those of you that are here with us all the time, but again, we like everybody to remember we're stronger together, get an idea of who's in the audience, how many other people are on the same treatment as you, or what you can look forward to, and then or see what's ahead. And also to give Dr. Wu, our guest this evening, an idea of who is here, how long since diagnosis.
Go for it.
Okay. And we have 12% of you are newbies, just diagnosed under a year. 15% of you are 1 to 2 years since diagnosis. 17% of you are 2 to 3. 15% of you are 3 to 4 years. 4 to 5 years is 17% of you. I'm in there too. 5 to 6 years, 11%. 2% of you have been diagnosed for 6 to 7 years. 5% of you, 7 to 8 years. 1%, 8 to 9 years. And 5% of you are 10 years or more. So thank you for being here and sharing the hope with us.
And back in 2020, when we started this program, there's two components to it. The Sunday ALK Talks, and then we also have a monthly Healing Arts calendar. And I want to basically say some gratitude to some of our instructors who are patients. Hanka Mosher, Eva Lamar, Heather Mayer, Nikki Hirsch, Megan Prose, who's a caregiver, Susan Remington, Tracy Cushing, David Cornbrooks, Dan Wilson, Amanda Stewart, and the person who I rely on the most to run these programs is Danny Carito. And I want to really say some gratitude, and I love you guys for helping us out. So I'm going to pass it over to Summer, who runs our Sunday Talks, who also does all of our programming with Ken Culver. So thank you.
Thanks so much, Mark. That is so true. These classes definitely help foster a sense of community, and it would not be possible without these volunteers. So we are so grateful for all of you. And we're always open to class suggestions. Also, you can always reach out to any of us on Facebook or email us at alktalk@alkpositive.org. Right, Dana? Um, I messed that up last week. Anyway, hello everyone. Welcome back. My name is Summer Farman. I have been an ALK positive patient since the summer of 2020. And am so glad that these programs exist to help make our community a little smarter and a little smaller. Tonight, we are honored to have with us Dr. Catherine Wu. She's a leading expert in cellular therapies and preventative cancer treatments. She serves as the chief of the Division of Stem Cell Transplant. Can I talk tonight, everybody? She serves as the chief of the Division of Stem Cell Transplantation and Cellular Therapies at Dana-Farber Cancer Institute and as a professor of medicine at Harvard Medical School. Her groundbreaking research focuses on developing innovative therapies, including cancer vaccines, to improve patient outcomes. Cancer vaccines are definitely what's on the horizon for us, and we are excited to have Dr. Wu with us tonight to help educate us and give us more information on where we are headed in this arena. So thank you so much, Dr. Wu.
Well, it's such a pleasure to be here, and wow, what a crowd for a Sunday night. This is pretty awesome. So it's great to see you and meet you, and it's an honor to be here. And Summer, that is a mouthful, so it's easy to trip over that. But I want to say one quick word about that, which is my clinical background is in stem cell transplant. Stem cell transplant was the original um, home of cancer immunotherapy before cancer immunotherapy became such a thing. So I think when I was in my late 20s, early 30s, I was drawn to it because it was clear that the curative power of transplant came from the immune response that our bodies were able to mobilize for patients with leukemia. And so my group has always been interested in trying to understand what the immune system is seeing and then how to mobilize that information so that we can actually crystallize into better cancer treatment. So it's a direct lineage, the cancer vaccines, from where we've been with stem cell transplant. So believe it or not, there is some tie-in together with the blood malignancies. I have a few slides for you, so I'll just go ahead and share them. Let me see if I can figure out this one. All right. Okay. So I'm gonna just give you a little bit of background and some of kind of the current work that we're doing, and then in the very end, how it relates to um, ALK tumors. And uh, I am very, very happy to answer any questions. So let me know if things are not clear or if I can explain things a little bit better. Okay, so I want to first start off by just saying that we are at a very exciting time of immunotherapy. By now, over the past decade, there's been so much progress that we realize that immunotherapy is a pillar of cancer treatment. At the center of this image is a T cell that is attacking a tumor cell. So the T cells in green. And what I want to say about T cells are the soldiers of our immune system, and they have a lot of different specialized functions, and they're listed here around the clock. And it is these specialized functions that have always made it a hope and a goal of trying to see if we could harness these activities of T cells in the service of eradicating cancer. And so it's not surprising that immunotherapy has kind of focused on the T cell activity. And it's also not, given that there's all these different types of functions, all the different types of immunotherapies that we have available to us now are really each and every one trying to harness a different aspect or different aspects of T cells. So you can see that there's many different types. They all do a little bit of a different thing. For today, I'm going to talk about vaccines. And I think it serves to kind of remind ourselves, what is a vaccine doing? What is a cancer vaccine? I think for better or for worse, we have all gone through the pandemic. So I think that the idea of a vaccine for infectious disease, so when you're a kid, there's all these childhood vaccines we get. And then later, as the pandemic happened, we got the COVID vaccine. So that is really to um, prevent one from getting an illness related to a pathogen. Cancer vaccines, for now, are different. They're what we call therapeutic vaccines. In other words, patients, for the most part, have cancer already. So what we're trying to do is to mount an immune response against determinants that are on the tumor cell and mount a response against the tumor cell. So what they share between a therapeutic and a prophylactic vaccine is they're both trying to mount immune responses in what we call an antigen-specific fashion. Antigens are what is expressed on the tumor cell or the infected cell, and mark those cells as targets of the immune system. So I think one of the things that's important to note about cancer vaccines is that a core essential function is that they're trying to amplify and diversify the anti-tumor response. So in this picture on the top part, what we show in the orange and circle and the triangle is that these are different, what we call tumor antigens. And if you vaccinate against these, then hopefully we can draw new immune responses that weren't there before. So these are the orange cells. Sometimes because tumor cells have these antigens, there can already be a pre-existing T cell response. The T cells may not be functioning great because obviously the cancer cells are growing in concert together with the coexistence of these T cells. But what we hope to do with the vaccination is to actually bring out the numbers of those T cells, but also amplify. So really expand the number of those soldiers that we're trying to get. Now, we talked in the beginning a little bit about all the different types of immunotherapies that are out there. This is a little bit of a complicated picture. Disregard the colors from the previous slide and only to note across the top here, these are kind of some of the different modalities of cancer immunotherapy you may have heard of. TIL therapy, immune checkpoint blockade antibodies, we have our vaccines, and then even gene therapy with TCRs, our CAR-Ts. And what this image is trying to show is, again, that idea in the pink, those pre-existing T cells, as opposed to green, the new ones that we're trying to bring in. Now, TIL therapy and immune checkpoint blockade, they really depend on what is already in the tumor bed in order for these therapies to do their work, right? Because TIL therapy is trying to get T cells that are already in the tumor, they try to pull them out and they reinfuse them. Immune checkpoint blockade is acting on what is already there. On the other spectrum, when you have gene therapy, you're trying to make a bigger, better, new sort of T cell. So that's adding, so these are completely new. They're what we call de novo T cells. What's special about vaccines, you can see, is that there's orange and there's green. You're really trying to access both these types of populations. And what happens when you do that, and that's on the title here, is that the other special function of vaccines is that they're trying to promote immune surveillance and memory. What does that mean? That means that if you have already been exposed to the tumor, but then you come in with a vaccine, hopefully you'll prevent that the T cells have that memory like, oh, the tumor should not be there. So they can kind of patrol and they also have memory that, oh, these, like I said, shouldn't be there. This is kind of a concept that is born out from infections as well. So there's some really neat anecdotes from even 200 years ago where patients who were exposed to measles at one point, fast forward 70 to years later, there was another outbreak on this island, and those who had gotten measles before did not get it again. The new cases were only those that had never been exposed before. So this kind of gives you an idea of the type of long-lasting memory and surveillance that can happen when you're adequately exposed to antigen in the right way. Okay, so those of you who kind of have followed the cancer vaccine literature, you'll know that this is not a new idea. It's been around for decades, but there's really been quite a roller coaster. Up and down, it's been popular. Some trial didn't work. It's been really down in the dumps. It's been all over the place. But I think that what's been exciting is that there has been, anytime you have progress, it really often is because there's been new transformative technologies that made once something that was not possible, possible. And for the vaccine field, I talked about antigens. The new kid on the block was neoantigens, and we'll talk about that briefly. But neoantigens, why did the current excitement about vaccines happen? Well, around 10 years ago, 10, 15 years ago, I would point to three important things that happened. So one is next generation sequencing became available. So suddenly people were able to kind of, at reasonable cost, on a tumor by tumor basis, find out what were the mutations that were present in a tumor. The second thing that happened is that there was better predictive computer algorithms. So if you had a sequence, the computer could say, well, what I know about the immune system and the way that it kind of encounters antigen is that I have a way of predicting whether or not uh, the antigen could even be seen by the immune response. And it's a little bit of a complicated concept, but I would just simply say that when I was a postdoc eons ago, we were only able to do prediction on a very limited, narrow subset of patients who have a very common HLA. HLA is the most complicated um, uh, part of our genome, actually. So what happened during the years between when I became a postdoc to the time that next generation sequencing happened was that actually there was a lot of data and people did a lot of training, and suddenly it was possible to predict not just the most common, but across the whole diversity and spectrum of people of various ancestries. So that was actually exciting. And then the third really important thing is that immune checkpoint blockade. So many of you have heard of anti-PD-1 therapy or anti-CTLA-4 antibody, Pembro, those types of drugs. They became they were FDA approvals that started around 2013. And why is immune checkpoint blockade important? Well, I think one thing to note is that this is the work that won the Nobel Prize back in 2018. So the work of Jim Allison for CTLA-4 and Hanjo in Japan for PD-1. There were many other people, our senior colleagues that are really wonderful who also worked on this and actually brought it to the clinic. So that's one thing to know. And I think the piece that was, I think, important for the vaccine field is the immune checkpoint blockade kind of at some level, there is analogies to Star Wars. So if you think about the cancer cells as a Death Star, I think all of you will remember that moment where Obi-Wan Kenobi kind of goes like, hmm, and the whole radar shield, the shield kind of comes down. And until that moment, you can't send the fighters in. So I think at some level, the immune checkpoint blockade is cancer's way of kind of shielding itself. And once these antibodies became available, suddenly it opened up a lot of new possibilities about how you could partner that together and send new fighters in to actually target the cancer cell. So I would say I would point to these three kind of important things that kind of came together. Now, what is a neoantigen? Well, I think we've known for decades now that cancer is a disease of mutations. And I think the important thing about next generation sequencing is it allowed us to go ahead and sequence the DNA of cancer cells and at some level unlock its secrets. We could find the mutations, we could figure out if the mutations were in particular genes that could tell us what were the cardinal pathways that particular cancer cells relied on in order to proliferate. So I think that's really important. But finding those mutations also gave us a new therapeutic opportunity because at least some of those mutations in the process of normal kind of going through the cell, some of them could be translated into peptides that were altered. So DNA becomes RNA, becomes peptide. Okay, so some of those peptides through the mutation become altered. And then again, in the normal process of what cells do, they take peptides, they break it down. And the way that the immune system is able to see it is that these peptides actually bind to HLA molecules. And that is how a T cell is able to see and sample on the surface whether or not an antigen is present or not. And so you can imagine that if there's a mutated peptide, well, those mutations, if it's recognized by a T cell, again, because the mutation is coming from the tumor cell, it is tumor specific. It's not on normal tissue. So if you have a T cell that's actually targeting this neoantigen, then it's only going to target the tumor cell. It won't hit normal tissue, so you shouldn't have any side effects. And so I think that's what was exciting. I think the idea of a neoantigen coming from a tumor cell, that wasn't a new idea. What was a new idea was that we had sequencing approaches that could allow us on an individual basis to discover what they could be when we put it together with those kind of predictive algorithms. And what we like about tumor neoantigens is because by virtue of coming from the mutations, they are exquisitely tumor specific. And because they're new events, they weren't there at at time of birth. They are new events, at least to the immune system. And so, like infectious agent, there's actually permissiveness in terms of the ability to generate a strong immune response. So you can generate a strong immune response, and they're tumor specific. And that's really different than where we've been in the past decades before we had these kind of sequencing abilities, where we were thinking about targets that were shared or overexpressed. Some of them are really good targets, but the reason why this circle is big is because they're variable in terms of the degree to which they're tumor specific, meaning there could be some cross-reactivity with normal tissue. And they are also variable in terms of how strong they are in terms of generating an immune response. And so, again, because we now have the sequencing ability, it becomes actually quite, it was very new when we first did this back 10 years ago, but by now it's pretty well accepted that we can take a tumor cell, we can compare it to a normal cell, you can sequence, you can compare, find out what those mutations are, and then bring this together with that prediction algorithm. And then what you spit out on the other side is a set of targets that are personal for the individual, that perhaps could be really good for immunotherapy targeting. So quite some time ago, and you can look at the date, 2013, we thought it would be really wonderful if we could somehow figure out how to directly collect tissue from patients, go through exactly that process. So mutation identification, HLA typing so that we could add that prediction algorithm in, and then come up with a delivery strategy. So back in 2013, again, before the COVID, before the pandemic, what was very well, there was a lot of experience in using synthetic peptides. So we came up with a way to prepare a vaccine that would reflect the menu of somatic mutations that was personal for an individual. And then recognizing that patients have lots of heterogeneity within their tumor, we wanted to launch a multi-pronged attack. So we were trying to go for 20 some different targets per patient that we could get in a syringe, and then combine it maybe with a strong kick in the butt sort of reagent, which we call adjuvant, to get the immune system revved up, and then maybe even put it together with immune checkpoint blockade antibody. And so I'm happy to say that in the red are the studies that we did. I don't think it's by chance that the first studies, not only our group, but also the Ugur Sahin, who oversees BioNTech, and also the group at WashU. I don't think it's by chance that the first setting was high-risk melanoma, because it's known that melanomas have a very high mutation rate. So for this type of genomics-based approach, it's nice to have lots of targets to start with, especially if you're embarking on a new type of study. Subsequently, we and others also tested this in newly diagnosed GBM. And the long and the short of it is that this kind of back of the envelope sort of idea that we sketched out, turns out it actually is safe and feasible. In fact, we're able to generate very strong immune responses. And actually, there's been examples of promising combination with immune checkpoint blockade. I'll give you just one example. So this was from our very first melanoma study. And these were two patients who had advanced disease. This is the months across the bottom, months after surgery. In the blue line is when they got their neoantigen vaccine. Because of the advanced stage of their disease, it wasn't such a big surprise in the red triangles that both of them recurred after the vaccine. But then their treating physicians started them on anti-PD-1 antibody. And you can see that what was surprising is that within 12 weeks, both patients went into a complete um, response that and then after a certain time, they were able to stop the anti-PD-1 therapy. And by now, this is something that we reported out quite some time ago. It's been at least 5 years since that time. And so you can see that these were patients at the time of vaccine with the arrows is where you see the recurrent disease. And then 12 weeks after initiating anti-PD-1 therapy, these have melted away. What was remarkable is that for this stage of disease, we know that anti-PD-1 does not work particularly well. So we do think it's the combination of vaccines, and I'm not going to go over it too much because we've done a lot of in-depth study of these patients. And what we do know is that at the time that they started anti-PD-1, we could already measure that there were neoantigen specific T cells at the site of tumor ready to go. So I think, again, I think one of the key messages is that the vaccines are able to elicit and draw out those elite T cell soldiers, and they stand at the ready. So when you come in with something like anti-PD-1 therapy, they can really go to town. This is just I was just trying to figure out a way to summarize a lot of the interesting studies that have been happening over the past 7 years. And the first two studies that from Sahin and ourselves are down here below. The left highway is for synthetic long peptides. The right-sided highway is for RNA. I'm going to go quick and just say that from my perspective, I think there are many ways to roam. I think that looking at the RNA studies that others have done and the synthetic long peptide studies that we have done, I think they both show activity. I think any therapy is going to need bells and whistles to make them work optimally. But I think in both settings, we've been able to see decreased recurrences after the vaccine, or as I just showed you, CRs when in combination with anti-PD-1 therapy. In both settings, there's been really nice studies basically using new technology that allows us to track T cells to the site of tumor, and we can see that the T cells are kind of coming in. I think another really interesting feature that we've been able to find is that after the vaccine, we see expansion of the immune response. So, for example, our vaccine targets at most 20 different targets. But after vaccine, we see many other targets that are specific to the malignancy also where there's an immune response. And that could have only happened if there was actually kill of the tumor, where there was a release of the intracellular contents of the tumor that became accessible to the immune system to mount new responses again. So I think this idea of kind of spreading the immune web so that there can be good immunosurveillance and prevent recurrence. And in fact, what's been super exciting in the last 2 years is that in one setting, the Memorial Sloan Kettering had a study in pancreatic cancer. We just had a study, it should be 2025, just come out in renal cell carcinoma, where we don't see recurrences in patients who have gotten these vaccines after their surgery or standard of care chemotherapy. But a lot of what's driving the current excitement in the field, I will say that these studies up until the most recent one on the Pinnacle are all academic trials for the most part. So they've been studies of like 10, 15, 20 patients. Maybe some of these studies are a little bit larger. The epitope spreading, that was more, I think, a 50 patient study. But what's been exciting is that last year, Jeff Weber reported clinical benefit in the first randomized open label phase 2 trial setting for melanoma. And that's what is shown here, that there was evidence that there was a relapse-free survival benefit of patients who got vaccine with Pembro. So that's the anti-PD-1. This is in the red. compared to receiving pembro alone. That's in the blue. So if you're kind of a higher on the curve, you're doing better. And you can see that this was a treatment of 150 patients. So this was with the support of our industry partners that this trial was able to be done. And then what's like really exciting right now is that there are large scale studies that are on deck. Some of them are enrolling or completed enrollment or starting soon. And just look at these numbers. So we're getting to hundreds to, if not greater than thousands of participants. So I think this is where we're going to be able to see if there's actually clinical benefit to the vaccine. So with all of this activity, are we done? What more do we need to do? And I would argue that we actually still have a lot of work ahead of us. My colleague, Ed Fritsch, we wrote this review together, and he absolutely loves the Golden Gate Bridge, as do I. So we had to throw in this figure in our manuscript, and you can see that the pillars have the different critical ingredients that go into a vaccine. But I think that what we show is that actually the cancer does create troubled waters that we have to bridge. And I think that there's a lot that we don't know, especially about the immune microenvironment. So in each patient cancer that we're trying to send these vaccines to and trying to generate T cells, what is the hostile environment that those T cells are entering, and how can we best optimize our vaccines to most effectively get our soldiers in there? This has been our paradigm at Dana-Farber, and it's the paradigm shared by many translational investigators. And it's the idea of starting at the bedside. So from our patients and the clinical trials we do, what are the insights that we can learn? What do we take back to the bench? And how does that kind of inform us of how do we go back again to the bedside so we can iterate and actually do better? And so what that means is that we do care about kind of creating more and more informative, small academic trials where we can test questions and really ask what are the next things that we need to learn. Because these are investigator-initiated studies, we are very grateful to our patients who partner with us and generously provide us permission to study their biopsies or to collect blood so that we can actually really do disease-specific research, so we can understand exactly how we can do better and look for those clues. And again, because we're working with our patients, every single tissue is very precious. And so we are always interested in learning about how to build new capabilities so we can maximize our learnings. So I'm going to take you through some little vignettes of kind of the things that we've been working on and things that we've been concerned and interested in. So first, in the subject of ongoing trials, I've been very fortunate at the Dana-Farber to work with wonderful clinical investigators in a variety of different areas. We have been partnering together with Alice Shaw and others in the Breakthrough Cancers group to try to see if we can initiate something in the ALK space. But for the time being, this is the menu that we have so far. And what I wanted to share with you was work that we did most recently in renal cell carcinoma, just to give you a flavor of what we've been doing. We reported out last month. And the way that we designed the study is our question was, well, it's standard of care for these patients with stage 3, 4 disease to go through surgery. But 30 to 50% of them are expected to recur in 2 years. So can we help them and prevent recurrence? And so again, we went through our regular, so we performed sequencing, we did prediction, we identified targets, we generated peptides that we put into these different vials, and then we introduced the vaccine to our patients. They're given in what we call a prime boost schedule. So priming is high antigenic exposure, so frequent vaccines within the first 3 to 4 weeks. And that's meant to kind of expose the immune system repeatedly to the antigen, and then we do boosting, and that is to stimulate immunologic memory. And this is what we think happens and mimics kind of what happens with a viral infection, for example. So we're basically trying to mimic kind of our body's natural physiologic sort of response to something like infectious pathogen. And so what was exciting about the study is in the 9 patients that we looked at, following them up to 40 months, that there were no relapses related to the disease that we saw. So this was, I just mentioned that 30 to 50% expected to recur at 2 years. So we would have thought that close to half of these patients would have had a recurrence, and none of these patients recurred. So very exciting. The other thing we saw was that there were very strong immune responses. So our workhorse assay is something we call an ELISpot. So what that is that we take patient blood, we put those blood cells in a small culture dish. We also put in the peptide that corresponds to the neoantigen. And if there is a response, those T cells in real time start to secrete interferon that we can capture. So each spot is truly one T cell that is kind of like saying, hey, I recognize this peptide and I'm generating a response. And so what's interesting is that the VHL mute peptide for a renal cell is like your ALK peptide. It's the same sort of idea. This is an essential RCC driver for which this particular patient had a mutation that we could generate an immune response against. So you can see this very strong response. On the bottom is what we call a positive control. So if this isn't positive, then there's something wrong with the assay. So positive is positive, check. The 2 negatives are negative, check. So the top one is our experimental. So it's confirming that 16 weeks after we initiate the vaccine, we in fact do see an immune response across the patients. There were a number of other kidney cancer driver mutations that were incorporated in the patient's vaccines. And we could, again, in the dark red, show that they were immunogenic. And then what was also very exciting for us, because I think it's one thing to generate a peptide immune response, it's another thing to confirm that these are actually peptides that are present on the patient's tumor, because we want the tumor kill to happen. And so what's nice about this is that we could show that actually there was actually, you see spots here, so there was definitely an immune response against the patient's own tumor cells. So that was very nice to see. And this is across the collection we could confirm in our laboratory assay. So just to summarize that study, I think what's exciting about that study is that it does show that we can prolong recurrence-free survival. We definitely saw strong, consistent immune responses including against mutated drivers and against the patient's own tumor cells in all patients. I think the important thing to say about RCC that's different than melanoma is it is what we call a lower mutation burden tumor. So it's not like melanoma. It is a more challenging setting to create a personalized vaccine, and yet we were successful. That being said, we have definitely more opportunities that are needed to find other classes of neoantigens that we can get at. So in other words, the same type of antigens that we can discover in melanoma are probably not the same in kidney cancer. And that's probably going to be true across the board. I think actually lung cancer is going to be closer to melanoma than kidney cancer. And that said, I showed you the large scale studies that are underway right now. So it'll probably be another 2 to 3 years to see the results of those kind of large scale studies to see if incorporation of vaccines will be practice changing. So that's a period of time where I think we have our work cut out for us. In other words, trying to innovate and iterate during that kind of observation time. As I said, we're working on disease-specific research. We're also thinking about building new capabilities. In terms of disease-specific research, some of the questions we're asking are, how can we define predictors of response? Can we better understand the tumor microenvironment? Again, we're sending these vaccines into patients who have had disease. So how do we make sure that the immune system is as robust as it can be? Can we understand the specificity of those T cells that are hanging out within the tumor? And this is a direct line to thinking about TCR therapy or cell therapy. Can we discover new antigen targets, as I just said? On the other side, in terms of building new capabilities, one tool that we often use is mass spectrometry. Some of you may have heard of that before. It's a pretty fancy machine. It allows us to directly see what is on the surface of a tumor cell in terms of peptides. Its downside is it's not always sensitive. So we have to work pretty hard, but we're getting better and better all the time. And that kind of provides us with more confirmation, and it allows us to discover new targets to go after. We talked a little bit about prediction, and the predictive algorithms are working pretty well, but actually I really want them to work 100% well. So that means we have, or again, our work cut out for us. And then the other thing that's really exciting about the current day and age is that we are getting really into these capabilities where we can actually visualize within a tissue how different cell populations are organized, so spatially within the tissue, and how can that inform us? So after vaccination compared to before, how are those T cells, how are they organizing themselves in opposition to the tumor cells? Are they getting close enough? Are they hanging? What crowd are those T cells hanging out with? Can we actually see that? Okay, so here's what about for this group, the ALK positive tumors. And have you had Roberto Charlie talk to you guys? He's a wonderful partner, and we've known each other for more than a decade now. So I think a lot of the things on the slide you guys already know, but I'll just for completeness go over it. So I think what is interesting about ALK is that it itself is an oncogene. It is what is driving the ALK positive tumor cells to survive. That means because it's essential, tumors are pretty sneaky and they can find ways to avoid the immune response. But ALK, they're never going to let go of that because that's what's actually essential for the ALK positive tumor survival. It turns out that ALK is not only highly expressed and selectively expressed on the tumor cells. By virtue of being a fusion, it is a kind of neoantigen by itself, but also it seems to be immunogenic. And so Dr. Charlie has done a lot of work in animal models that have demonstrated that this peptide by itself is pretty immunogenic and it can generate anti-tumor activity. We've worked together with him over the years to work with some of his mouse studies and make sure that it's relevant to the human side. And it turns out that we can also identify immune stimulatory human peptides. And in some human patients, there is spontaneous immunogenicity that's identified. So it makes a lot of sense to think about how we could generate ALK peptide that could then stimulate a T cell that could go after the tumor cell. This is the work that we did together a while ago. This was originally using mass spec to find those peptides and confirming that it would work for peptide vaccination in an animal model. This is cute, his little stop sign, the go. And again, so this is the work that we did. So what we can do is you can take tumor cells, maybe I won't go over this, only just to say that um, we're trying to strip the tumor cells of their HLA molecules and peptides, and then we run it through a mass spec and see if we can identify the peptides. And if you look at peptide number 2, in the examples that we ran, we could definitely detect the peptides that we predicted. And so that was actually pretty exciting. So that's the last slide I have by ALK, and I'm happy to talk about it more. I would say that with breakthrough cancer and with Dr. Charley, we are trying to figure out how to get going with a vaccine study. I know Dr. Charley is also interested in TCR therapy. In both of those cases, what is reassuring is that we do have a peptide to go after. Um, some of our challenges that we're hopefully quickly working through. Number one is trying to figure out, in one of the slides, I talked about mRNA versus peptide. So we want to sort out, do we want to do one? Do we want to do two? Do we want to do both and compare them in different arms? So we're still working through some of the details of the design. This is also with Dr. Shah. And the other piece, true for any study, is that we have to secure the funding in order to make such a study feasible. So that's just the brief update that I have for you. I think I just have a few more slides and just maybe so you can kind of get where I'm coming from. So I think in terms of how do we move this through, I think I'm really I have a lot of gratitude for my wonderful trainees. So the MDs and the MD PhDs and PhDs that come to the lab and put their heart and soul into working on the vaccines and trying to do all the things that I talked to you about before. So I think training of young people is super important. This is this correspondence that I've been having with an 8-year-old gal who's the daughter of a faculty member. And she said, "Dear Kathy, I think I have a cure for cancer. Do you know about the coronavirus? And like, could you do this? And could you do that? Da, 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 da." And in the end, she says, "I also think you should get it through a shot. Love, Eleanor." So we've gone back and forth a few times. And I do think we have like a budding young future visionary scientist coming through the ranks. One of the questions I get asked a lot, which is, how do the vaccines fit in with cancer treatment? And I would say that actually they make a lot of sense in every single phase of disease. And what do I mean by that? Well, as an oncologist, I think we're always dealing with patients who already have malignant cells, tumor. And so just as I mentioned to you in that example of the stage 4 melanoma, these are people who actively need treatment. For the immune system, it's a pretty high bar. We know that tumor cells are doing everything they can to kind of shut the immune system down. And what that means is that we really have to think about what is the best combination. The vaccine is, I have high doubts that the vaccine by itself can do all the heavy lifting. It needs some synergy together with other agents. And I think this is where combination with TKIs or therapies, so long as they're not bad for the immune system, so that you can decrease the tumor burden, makes a lot of sense. The recent pancreatic cancer trial the vaccine was given to patients after standard of care chemotherapy, and that seemed to work well, at least for 50% of the patients. So I think it's possible. And we do have some trials that we're working through in patients with advanced disease. So I think that's an area that we need to continually iterate and think about what's the right combination. I think probably the clinical successes we've had so far, like the kidney cancer study that I just mentioned to you, that's in the adjuvant setting. So that's to prevent relapse. Makes sense that it's working well because you've already cytoreduced the disease. The immune system is always going to work best when there's not active disease going on. or just minimal. So again, I think that will always be, in my mind, a very encouraging place for a vaccine, um, either vaccine alone or also in combination. So combinations are still important. I think probably the greatest gains that we will all collectively have is in cancer prevention. But that is challenging in the sense that we really have to kind of figure out the cost and time of manufacture uh, challenge. And this is, again, something that my group is actively working on so that we can, we have to figure out a way to decrease the actual cost of goods of such a laborious approach like a personalized vaccine to be able to make it available to larger patient populations. And I think this also gets to the idea of trying to smartly couple together what we know about the patient's own disease, the host immune response, and what does that mean in terms of trying to build the right sequence of modalities that we're trying to uh, select on a person by person basis. And I think this is my final slide, which is that I do think that as we kind of enter this age in which I don't think we're too far along from the time that genome sequencing should be part of the clinical test that patients get when they are first diagnosed with cancer. So if we're going through all of that and finding mutations, and why shouldn't it be that we can't actually order up our personal cancer vaccine through our pharmacy and then to be able to administer to our patients within a couple weeks. So this is aspirational, but this is what I think could and should happen. So I want to thank you for your attention. I've received a lot of support from different foundations and from NIH. And I want to acknowledge that it's not just me. This is the brain trust that I have. So Nir Hacohen, Ed Fritsch, and Patrick Ott have been really my partners throughout this whole 10-year plus odyssey. But really, our whole infrastructure at the Brigham, at the Dana-Farber, on the clinical side, from the immunologists, also our proximity to the Broad Institute and all the fantastic technologies that we have available and the individuals in so many different departments, ranging from basic fundamental RNA biology to single cell technology, imaging. And then I mentioned our trainees who are just amazing. And it's really our young people who help to really drive this forward. So I think I just want to make sure that you know that it's a whole force of people who are working behind the scenes on all of this. So I'll stop there, and I really welcome any question.
Thank you, Catherine. Ken Culver with ALK Positive. Thank you again that was really, really fantastic. And I wanted to say to everybody, if it's confusing to you, it should be, because this is a very complicated area of medicine and science. And the fact that there aren't any approved cancer therapy vaccines, really, that tells you that it's tough. And I wanted to just answer a couple of the text questions before Mark and Summer. First of all, neoantigen. I know Catherine said it, but I just wanted to make sure. Neoantigen just means new antigen. It's something new made in the tumor. So therefore, it's an ideal target because it's only going to be in the tumor. There are quite a few neoantigen vaccine trials available, some of which are currently allowing ALK positive lung cancer patients. There are also a number of companies who will make you your own specific neoantigen vaccine. They run around, the companies we've spoken to in the medical committee, they run around $100,000 each.
Mm-hmm.
But one thing to keep in mind, and I'd love it if Catherine could comment on just this in general, is the computer algorithms that are used to figure out what your neoantigens are in your tumor are not the same. The companies are using different ones. They may or may not be finding the neoantigens that are most likely to induce an immune response that will kill your cancer, create this memory that's so important. So we are at a learning stage, and we in the ALK Positive medical committees are talking with investigators and talking with companies and tracking this along, looking for the first sign that there's a breakthrough for our patient community. And as Catherine said, our investment in BTC is really a large way addressing this need. We've got people like Catherine who are part of it. Vincent Lamb, who's working on vaccines, is a part of it. We're hoping that that group is going to bring together a group of trials that are going to be able now to start to dissect out and have effective vaccines for ALK people like you on the call who are in the MRD state with ALK positive lung cancer. So those were just a couple of the general comments I wanted to make. Summer, Mark, I'm sure I skipped over the most important questions.
I think, first of all, I want to thank Dr. Wu, but I think that the combination therapies is people are still going to have to take their TKIs with this combination of a vaccination. And can you address, in the summit, are you going to address any of this in the summit?
During our medical presentation of the summit, we will be talking about our initiatives to learn more about vaccines, and we're going to have the BTC group there who will be talking about moving forward. And Dr. Chiarli's vaccine is certainly a key part of that. Yes.
Thanks. Summer, did you see any chat questions that you
I
want to
did see some
address?
interesting ones. Dr. Wu, this was great. Your slides were so clear. You helped break down information for us. Of course, it is still very overwhelming and difficult to dissect and grasp, but still just laying some of that out for us. And it'll live here as a reference that we can go back just to understand where the direction of personalized medicine and in general, just giving us hope for the future for sure. So thank you so much for that. I thought this was a great question. Why does my immune system need a vaccine to train it? I have all of these billions of cancer cells for several years. Why isn't my immune system attacking?
I think that's a wonderful question. And there's a slide that I didn't show. But basically, one of the fundamental characteristics of tumor cells, and this is, the mutations help with that, is that they are always evolving. They're changing. So they can acquire new mutations. We know by now that even in an individual that there may be a mix of populations, some with higher levels of some mutations or lower levels. So what we call heterogeneity does is that whenever there's some pressure on the tumor, it's a little bit like whack-a-mole. They kind of turn this population off, but then this other population grows. And one of the also cardinal things that cancer cells do is that they get wise and they figure out how to turn the immune system off. So probably before we realize that there is a diagnosis of cancer, there's probably already a behind the scenes battle kind of going on where the immune system is actually what we call equilibrium, trying to keep the cancer at bay. But the kinetic of the cancer cells is that they keep growing and evolving. So they'll kind of find a way around it. So by the time, unfortunately for our patients with cancer, they have already evolved to the point that the immune system doesn't work and doesn't function as well. We see a bit of that evidence of that archaeology when we start to analyze in our patients the specificity and the immune health from patient biopsies. So we do see evidence that probably in the past, there were actually the right soldiers that were there. They probably for a while kept things at bay, but eventually, by the time we see those T cells, they are what we call exhausted. They have markers that show that they are exhausted, that they've been in the battle, and they weren't able to hold it together any longer.
That is a great explanation. Thank you so much, and great question. Where I had, oh, combination, it would obviously, I feel not obvious, but I feel like from what I have learned and others that it does seem that combination therapy is the way of the future too. That we can't, combat ALK with one single agent. So somebody had said, would we continue to take our targeted therapies also?
Yeah, I think that's also a really great question. I think that the real truth is we don't know. So I gave an example in melanoma where the patients got their vaccine and they had anti-PD-1 therapy, and at this point, those individuals have been off all therapy for a number of years. So I think it depends on how strong an immune response you're able to generate. If there is really absolute no evidence of disease anywhere, I think it ends up being a clinical judgment because no one really knows. And yeah, and then the other aspect is to try for a time limited amount of time, and and the good thing about ALK is you actually have something to monitor, so you could figure out if there was any change in the dynamics over time.
In the past, we've heard ALK has cold cells versus a hot cell with other cancers, like a different type of lung cancer. Do you find that that would be still difficult to do with the modern technology, or do you still think that our cells can be more difficult to turn on?
Again, I think that's one of the features that I think makes me enthusiastic about the vaccine is that it's able to make cold tumors at least warmer. So, now how consistently do we see that? The Where we have seen this is in the setting of glioblastoma, which is a notoriously cold tumor that we can stain slides afterwards, and we can actually count the number of cells, and
Mhm.
and and the numbers do rise after vaccine. Other interventions can do it too. So, for example, radiation can do that as well. So, I think that there are different modalities that can, like I said, warm up the environment.
Mhm.
And then the question becomes, what is the right combination for ALK? Is vaccine alone enough? Do you need the TKI to help? What is that? Going back to Star Wars. So what's gonna bring the shield down
Yeah.
to let the T cells in?
I just want to mention to the others that are out there that we've had Dr. Killoran twice and Dr. Lamb on in regards to vaccinations. And this is what we've been trying to do for a long period of time. And it's exciting that we're almost there with some type of breakthrough. So we're super excited. And we really appreciate you coming in and spend your Sunday with us and giving us a slideshow that brings us more knowledge, and knowledge is power. So we really appreciate it. Somebody have any closing remarks for Dr.
And I
Wu?
did. I just wanted to ask a couple, one more actually. I can, we could just keep you here all night. So, like Mark said, we do want to be respectful of your time. There were so many great questions, though. But I guess, do you feel like with the current stances politically, kind of on anti-vax and funding cuts, that this will slow down research at all? We were kind of talking prior that it takes a lot of private funding, and any
I mean,
comments?
I won't lie, it's a very stressful time to feel that you're under siege for work that should really be very neutral, that would be beneficial to all. Um, uh, and I think um, I think our infectious disease colleagues are bearing the brunt of a lot of attack. So just going by the headlines among universities, Columbia is bearing the brunt. I think among NIH funding, the NIAID is bearing a disproportionate degree of animus. Which I find ironic because it was really vaccine that got us out of the pandemic. Otherwise, we'd still be doing a lot of lockdown activity. So I find it very ironic. It is challenging. I know that a lot of the companies have scrubbed the word vaccine from their the nomenclature of their therapy. I kind of want to stick with the vaccine just because maybe I'm a contrarian. But because it is what it is, it's doing what it's doing. I think the federal funding will be challenging. I think it's going to be tricky times ahead of us. I haven't ever seen anything like this before. So I think now more than ever, I think we are grateful for support from foundations and philanthropy, and we're trying to figure out how to diversify. All of us get a lot of NIH funding, and it really helps. And I think the part that kind of cuts to the quick the most is I think it is disproportionately affecting the young. So I think already there's been spots cut from graduate programs. Young 22-year-olds, these are the ones who experienced the pandemic when they were freshmen in college. I know because I have one of them in my own household. So they kind of survived the pandemic, and then now as they're trying to finally get footing and trying to think about this, a lot of them are ready to commit to graduate programs and really enter in this profession. They are finding a lot of uncertainty and lack of programs that are available to train. That's also true for some of my extraordinary young scientists who are going on the job market where there aren't jobs right now because no one is actually I think the universities are not in a position to hire right now with so much uncertainty. So it's pretty challenging.
Yeah,
But
and you showed your slide with all your young trainees and how they're helping to
they're
change the
wonderful.
landscape. Yeah.
They're wonderful, and they're the energy, and they're really the stars. So the part that is tough for me to see is that I think they cost the least, and they're being affected very hard across the board right now. Yeah.
Well,
Sorry.
no, we want to keep it real here at ALK
Yeah.
Talk. All about educating, and knowledge is power, whether that knowledge is, positive or negative. So we appreciate you being real with us and for sharing your Sunday with us. And if you look in the comments, which I can send to you, everyone is so grateful. It was a fabulous presentation, and you really taught us so much. So thank you so much for joining us tonight. And if everyone would like to unmute and say thank you, and we will see you back next Sunday for a talk with Dr. Laura Petrillo on palliative care. And what exactly that looks like. So thank you all for joining us. And thank you.
Thank you so
Thank you,
much.
Dr. Laura.
Thank you.
Thank you so
Thank
much.
you so much.
Great presentation.
Thank you.
Thank you.
Take care.
Thank you.
Thank you so much.
Explainer in chief.